first things first, DNA is deoxyribonucleic acid. Deoxyribonucleic acid. There finally is for you. Deoxyribonucleic acid. You might have to write that down. Do you have to spell it correctly? Absolutely not. But you can see where DNA comes from. DNA. Grand. Now, what do we need to know about DNA? Well, we actually have already learned this, but DNA is located in the nucleus. How is it located in the nucleus? Well, because DNA and protein make up chromosomes, which is the same as chromatin. Okay, and that's in the nucleus. Okay, and that's basically what the whole last chapter was about. It is double-stranded, or it has a double helix shape. Okay, so let me write that up there. Double helix. Now we'll see exactly what that means, but double helix. Grand. DNA plus protein is chromatin, as we know, great, and it is hereditary. Okay, what does that mean? It means you get it from your parents. Now, even if you get DNA or genes that your parents don't seem to express, so say you might have a different hair colour or a different eye colour to them, you still got the DNA from them. You still got the genes from them. They just didn't express it. They got it from their ancestors. So it could be their mum or dad, or their mum or dad's dad, you know what I mean? So it can be carried through. So sometimes it can skip generations and generations, and randomly then you've got a trait expressed that none of the parents have, or the grandparents even. Okay, so it's hereditary. Now, it's also important to note, and we've mentioned this before, we've definitely mentioned this before, DNA is also found in mitochondria and chloroplasts. So I'm just going to write mitochondria here, but I'll just remind you, in terms of the mitochondria, we said you get the mitochondrial DNA from your mother. Okay, so just if you can't remember that, go back to the cell chapter and look at where we explain that out. Okay, so the structure of DNA. Again, we've said this is double helix. So what that means, and I always mess up my diagram with this here, because I do too many wiggles around. But that's one set of chromosomes, or that's one chromosome. If you want to remember, there's two sets of chromosomes. And this is the other one, here. They are intertwined in each other like a lace, like you got two hairs and wrap them up. Or just if you unraveled a piece of wool, you'd see it's made up of little pieces. That's called double helix. Okay, so that's what we spoke about there. Okay, so we've got gene sections, coding structures make up 3% of DNA. So your DNA, 3% of it is genes that code for proteins. So anything that's made in your body is a code for a protein, as we will see in this chapter finally. And it will finally come to fruition for one of the last times why protein is not just a food, but how your genes actually work. It says junk DNA or non-coding structures make up the rest. Now, the weird thing about this is extremely outdated. So much so that the examiner, remember the examiner before had to put on stem cell research for tissue culture. Junk DNA, they actually realised since this is not actually junk, it actually does stuff, like a lot of stuff. And that's why they call it non-coding DNA as well. So that's the same thing. And they actually had to put that on the exam. So that's why I have it in the notes there. So 3% makes up gene coding, so we'll just say coding and non-coding, right? So coding for proteins, and then non-coding. And again, humans don't massively understand that. They're only recently, and this is in the last number of weeks, by the way, this is in the last number of weeks, they've only recently discovered that they think, and this is going to go against everything you believe in science, everything you've learned since first year. You know, in first year, you probably learned in science that you have inherited characteristics and non-inherited characteristics. So, for example, an inherited characteristics would be your eye colour, and non-inherited characteristics would be you can speak French. They're only starting to realise that the things you would do in your life that are non-inherited characteristics can be passed on to your offspring. They have no idea how. They have no idea where it's stored. Now, I'm not saying you wake up as a baby and you know French, but there are certain characteristics that they've got. And if you wanted to have a look at it, fair enough, have a look at it. But they believe it's something to do with this non-coding DNA, which they also feel does a lot more than just this code for protein stuff here. Anyway, what actually happens though is on this stuff here, on this structure with DNA, we're going to deal with what's known as 4 bases. We've got A, T, G, and C. A, T, G, and C. Now that stands for adenine, thymine, guanine, and cytosine. I'd suggest it's probably good for you to know those words, but most of the time you don't need them. You can just go ahead with A, T, G, C. These are bases, and what these bases do is they actually lock one strand to the other. That's what we're going to talk about for now. They lock one strand to the other, but at the same time, they're the instructions for the making of a protein, as we'll see. But at the moment, they lock one strand to another. Because as you can see from our diagram in there, they bond to another base. They bind to another base. So A, T, G, and C. On the top of the next page, you will see, or you can see in your diagram on the bottom of page 24, that A and T bind together and G and C bind together. So adenine always binds with thymine and vice versa, and G always binds with C, and C always binds with G, and that is in DNA. Remember, this is DNA that we're looking at here. Now, what I've tried to do is show you how this works in one diagram. So the next probably, 260 seconds are going to be a little bit confusing. So if you're kind of feeling yourself drifting off a little bit, I'd pause it now, take a few deep breaths, go for a walk, just around your house, or do whatever, and then come back to this, because this next part here is going to be where you either get it or you're left behind. Even if you wanted to take down this time period in the video so you can come back to it if it slips your mind and you need to just watch this part again. Okay, so on the next page, on page 25, I've drawn a structure. I've gotten a structure up here that explains everything you need to know. You'll never have to draw this structure, but it's all in one diagram. So I wanted to put everything into one diagram that we needed to know, but I also want to make sure that you know you don't have to draw these pieces here. Okay, now if I look at my double helix up the top, okay, the blue strand has certain bases here, and the red strand, the black strand has certain bases here. Now, the colours I've just put there just to show you there's different colours, but it doesn't make too much of a difference. Okay, so they bind together. So if this top red part here is A, that part has to be T. If this part is C, that part has to be G, and so on and so on. They match up on each other. If I was just to take one bit here, so one bit of the DNA and one base, so one bit of that blue strand and the base, which equivalently from my diagram would look like this. Okay, and this is not how it looks in real life. It looks like this. That is going to make up what we are going to call a nucleotide. And chemically, we have to be able to draw what a nucleotide is. This is the only chemical part that we need to know. So a nucleotide is the subunit of a DNA. When you have loads of these nucleotides, they make up this DNA strand. Okay, this chromosome strand here. Okay, so hopefully that makes sense. But this is the subunit. What it actually is made of is a phosphate. Okay, so you don't worry about the way they've drawn it here with all these angles, but I'm going to try stick to it a little bit here. So phosphate, a deoxyribose sugar. So that makes sense. Deoxyribonucleic acid, deoxyribose. I'm just going to write sugar here. Okay, so D, sugar. And then a nitrogen base. And this nitrogen base is what's going to change. These two are always going to be the exact same, whereas this base will change. Like it just says base down there, but that base could be A, T, G, or C. So I'm just going to write A here. That is a nucleotide. Now, that nucleotide will be underneath, bound to another nucleotide. So, for example, they had A in their diagram there. The next one is they've put down as G. Okay, so how that works is that bonds to that phosphate here. Grand. That phosphate still again is bound to this deoxyribose sugar. Grand. And then that is bound to a base. What was the base here? That was G. Okay, cool. G. And as we keep going down, if I had loads of space and I could keep going down here, that would actually be what we are showing up here with this strand with the red parts coming out of it. Okay? Now, if we were going to do it here for the other side, remember this base has to bind to another base over here. So if this is A, this base here has to be T. Has to be T. And what happens on the other strand is the exact same. So we've got the DS here, and we go up to this P here, and that goes out to the P here, and that P goes down to the sugar here, so the DS there, and this base is important because G binds with C. Okay, so individually, that thing there, those three things there are a nucleotide made up of a phosphate, a deoxyribose sugar, and a base of whatever base it is. So that's a nucleotide, that's a nucleotide, that's a nucleotide, that's a nucleotide. If we kept going up or down here, that's going to form a strand, which is, say, the blue strand here. And those strands bind together in DNA to form that double helix structure. That looks like that intertwined piece there. Okay, so a little bit strange, worth maybe taking your time just to kind of make sure you're alright with that. How they bind together is via hydrogen bonds. Now again, I am no chemistry teacher. You probably know a fair amount more about chemistry in terms of Leaving Cert than I know. I know loads about junior cert chemistry, which isn't a hell of a lot, I'll just tell you the truth now. Okay, being honest. But now here, in terms of these hydrogen bonds, if you ask me how they work, I could kind of tell you, but if you ask me from a biological exam point of view how they work, I don't care, okay? Because we don't need to know. What happens here is there's A and T form two weak hydrogen bonds. So, look, there's how we are drawing the hydrogen bonds, okay? Two weak hydrogen bonds, which is fantastic. Great. G and C form three hydrogen bonds. So 1, 2, 3. Great. So hydrogen bonds. Fine. There is one last thing we need to be aware of. One last thing. Okay? We need to know that A and G are considered the purines. So these two here, and it's not written actually on that diagram, it's just in your notes, so feel free to write it in. A and G are the purines, whereas T and C are what's known as the pyrimidines. And God knows if I spell that right. So those two down here are the purines. In terms of the bases, they are the pyrimidines. Okay, so be careful, because this A always binds with this and G always binds with this in DNA, but A and G are purines and T and C are pyrimidines. So don't get that mixed up. Definitely worth your while spending a little bit of time making sure you're okay with that. And that's DNA, all in this diagram here. So there's our, this is going to probably make it look real messy, so we'll put it here. There's our nucleotide, which consists of a phosphate, a deoxyribose, and a base. We have 4 bases. We know they bond with a specific thing. So A with T, G with C, grand. We know that there is a two weak hydrogen bonds between A and T, and then 3 hydrogen bonds between G and C, grand. And we know that the purines are A and G, and the pyrimidines are T and C. What do they mean? Absolutely no idea. But you definitely need to know that category and that grouping. Fine, that's DNA. Not that bad. Not that bad.